Hello, everyone. I'm Nicola Bordin, and I'm a research fellow at University College London. Today, I will tell you all about how we're scaling up protein classification using CATH-AlphaFlow and Chainsaw, which are two new tools that we developed for the CATH protein classification resource. So, what is CATH, you wonder? So, CATH it's a resource that started in the early 90s, and over a period of over 25 years, we classified protein structure domains from the protein data bank, and we curated their assignments into homologous superfamilies, as in every single member in the superfamily has a common evolutionary ancestor. So this is an overview of how we do it every week. We take new structures from the protein data bank, and using a variety of tools, both external and internal, we identify domain boundaries, we chop them into globular domains, we assess their quality, and then we assign them to CATH superfamilies. Most of these assignments are done automatically by a variety of different pipelines that we built in-house. But whenever we encounter some very difficult cases, we rely on some experts in our team that are very good at manual creation for protein assignments. So over these 25 years, CATH grew steadily and happily, I would say, alongside the protein data bank. But the rate of deposition of protein structures got quickly taken over by protein sequences. And now the number of sequences deposited in protein sequence resources such as Tremble, SwissProt, or Magnify, now counts to billions and billions of sequences. So in order to take advantage of this information, kind of encoded in sequence data, every 2 years, we freeze the CATH-B release, which is the weekly release of CATH data, and we create a new database called CATH Plus that includes a lot more information. So we have curation, we subclassify proteins into functional families, where every single protein in these functional families, they share the same function, and then we make this data available through HMM libraries and other data sets. But in order to do this, our first step is to take this evolutionary information that is encoded in our protein structures. We encode it into sequence profiles, and then using these sequence profiles, we build hidden Markov models, which we then use to scan against RefSeq, UniProt, and other sequence resources. By doing this, we're able to expand our knowledge of 450,000 domains, 500,000 now, to roughly 150 million domain assignments on UniProt alone. And the advantage of using UniProt in this case is that we're able to inherit their expert creation on these entries. So we're able to inherit Go information, EC terms, drag bank information, and so on. So this kind of continued for the past 10 years at this point, and this plot also changed very dramatically in 2020. I will show you how it is going to change later on with the release of AlphaFlow database. So their initial release covered 365,000 protein chain models for 21 model organisms. And then they had subsequent releases for very interesting subsets, sub-datasets of such as SwissProt and organisms causing neglected tropical diseases. But the latest release covers UniProt. So this has been a very interesting challenge for us on how to tackle this data. The first step, when we started looking at this initial release for 365,000 domains, protein structures, for 21 model organisms, it was to assess what was good enough to be comparable to an actual protein structure from PDB and something that looks like an actual globular domain that we classify in CATH. And we noticed from the get-go that since AlphaFold was trained on PDB, structures that resembled something that we already classified and was already available in PDB had a great quality with PLDDT, which is AlphaFold's metric for quality. It was very high. But then as soon as we move further away from what is known, like for very remote homologs or potentially new superfamilies, we noticed that not only the quality dropped quite significantly, but also that the disorder level found in these potentially new domains was also increasing quite a bit. But then once we started looking at these individual cases, we noticed that we had to remove some of these models because they had some problematic features that don't really belong in CATH as being a globular curated kind of domain classification. So we noticed that some of them, they have a high percentage of unordered regions. So we have roughly 60% of the domain or more that is highly unordered. We have some very long unordered regions at either end of the domain with very long stretches of kind of unordered regions. And then some other domains there, they only have a few secondary structures, so they're not really a domain in itself. And some others, they are poorly packed, so the core of this globular domain is not globular anymore and wouldn't really pack very well. So once we started kind of quantifying this for the whole first version of AlphaFlow, we noticed that roughly only 52% of models in the first release of AlphaFlow were deemed of good quality, for at least for CATH purposes. So the vast majority of domains that we discarded, they had an average PLDDT for the domain that was below 70. And some others, they showed these packing issues, or others that were very, very tiny in terms of having less than 3 secondary structure elements. So this was very interesting for us in a way, because once we started looking into this treasure trove of data, which accounted almost to 70% of CATH, so close to 15 years worth of structures, we found some new folds we haven't encountered before, and we made all this new data available, which made also some new discoveries possible. And then we made a combination of CATH and these new good models that we identified in AlphaFold available to search using these new and ultra-fast structural aligners such as FoldSeek. So while we were looking at the AlphaFold dataset, 93% of the good models, they entered CATH and they looked like something we already classified in the past. So in the past, we already had a superfamily assignment for these using HMMs or kind of a deep learning predictor. We noticed that there was still a portion of them, although tiny, that had potentially some new faults in it. So after we used some of our manual curators to look at this data, we identified 24 new faults in clusters that at least had a human protein. So you can see that from the ones on the left, the vast majority, they are alpha. They're very alpha preponderant. And the ones on the right, they're like kind of mixed alpha beta. There's still some very interesting new arrangements. You can see here, there is also a very peculiar hard domain on the top right corner. And since that data release, we also focused on kind of curating all the rest of these clusters that we identified that couldn't enter CATH in the past. But in this case, these are all the remaining ones that didn't have a human relative. And by doing so, we identified some more. So bringing in the total to 111 new faults. So this data now, you can download the whole of CATH and this particular assignments for the 21 model organisms in Zenodo. And you can read all of our work in the manuscript that I cited there. You can also go to the FoldSeek web server and search against CATH-50, which is a combination of the whole of CATH 4.3 plus all the good models that we identified clustered at 50% sequence identity. FoldSeek is extremely fast because you can also search one structure against our database, or you can download FoldSeek on your local machine and do all the searches yourself, especially if you need to do large scale analysis. But the issue we encountered after this is now that the plot is actually up to date, you can see that we have roughly the same number of structures for the same number of sequences that we have in Tremble. So even the previous protocol that we had in place wasn't really suitable for this. So we had to move to something slightly different. This is a picture here in the figure. You can see the protocol that we used for our analysis of 21 model organisms, which is a cascading approach that looks at first if we have some HMM matches, and then if we don't, then we try to predict the superfamily for PFAM domains, or what we call new FAM domains, where we don't have a CATH or PFAM assignment. we predict it using this new machine learning method called CATH. And then using the structural aligners, fold-seek, and also some slow but also very accurate ones, such as SAP, we're able to, using this homology-based cascading approach to bring in structures into CATH. But this method needed to had one main limitation. It needed to be kind of parallelizable. It needed to be parallelizable and scalable. So what we did over the past year, we turned CATH-assign into a scalable workflow, and for that we used Nextflow. And one advantage of this is that it's HPC laptop and infrastructure agnostic. During this ISMB, I'm going to actually give a tutorial on it, and everyone is going to be able to run it on a Google Colab notebook. And this combines CATH-assign and its philosophy with some new tools that we built in-house for this. And it's truly scalable and parallelizable thanks to Nextflow. So this is the worst case scenario for our HPC in our lab, where the smallest slot you can get on the HPC is one CPU, 1 gig of RAM. And we can run 100,000 structures directly from the Google Cloud Storage through chopping to a final summary of assignments in 10 hours. So once we start having more resources for this, we can actually try to tackle the whole of AlphaFold. One of the advantages of AlphaFlow, though, is that it's agnostic in a way. So it runs on PDB structures, it runs on ESM models, and it also runs on AlphaFold PDB and ESM models. And everything, it not only just relies on single domains assignments, but also can deal with multi-domain entries, such as the CATH KIMFAMs, which are our classification of protein kinases. The pipeline consists of multiple modules. You can run everything within it, from data retrieval to domain assignments, chopping using multiple tools, optimization of the boundaries, quality assessment, and all this reporting all in a single customizable workflow. And this is going to be what we're using. At the moment for tackling AlphaFold and ESM Atlas. So the last part of my talk is going to be on one of the tools that we built for CATH-AlphaFlow, which is called Chainsaw. So while some of the assignments, especially for domain boundaries, they were heavily relying on either homology information such as HMMs or these new embeddings-based predictors, we decided to build something new to deal with this data set. So here we present Chainsaw, which gets its name from being able to go over a chain and seeing domains and then chopping the domains, hence the punny name and being called Chainsaw. So Chainsaw is a 2D to 2D CNN with a ResNet style architecture based on Rosetta. And we take different input features, including an inter-residue distance matrix. We take two secondary structure co-membership Peterson matrix. We also use secondary structure boundary residues, and we combine all this information as the input features for the model. The model then kind of takes this matrix and then predicts a soft adjacency. And once it finds these boundaries, it goes into a set of recycles, and then we feed them back into the model in order to get a proper domain assignment at the end. So here's a picture of the actual model architecture, as you can see here. And once we tested it on multiple data sets, we benchmarked it on various CASP data sets, on CATH and on CATH assignments, and we noticed that it tends to outperform other state-of-the-art tools such as Unidoc and Pew, not only on the correct number of domains identified, so how many domains are within a protein chain, but also the domain boundary accuracy was higher overall. So these are some chopping examples. As you can see here, the ones on the left are PDB domains. So this is one of the advantages of not having to rely on PAE data, is that we can run it also on PDB structures. But the ones on the right are AlphaFold models. As you can see here, these are what we would call kind of problematic cases that will take some of our manual creators to have a look at. The top one on the top right This one is a multi-domain protein. It's a multi-domain chain, and you can see that it will be quite problematic also for a human curator to kind of find what are the exact boundaries for this, and the model does pretty well. Well, the one on the bottom right, it's even more tricky because it's consisting of two discontinuous domains. So as a final part is that whenever we have a look at AlphaFlow models in particularly, sometimes AlphaFlow doesn't predict well the whole structure. So you have some domains here and there where it was actually able to make a prediction interconnected by these various spaghetti-like regions. And Chainsaw is able to identify correctly the domain portions of it out of the structure, and it doesn't oversplit. As you can see here, for the same structure running on with Unidoc, you can see that the domain on the right it's split into multiple ones incorrectly. So both Chainsaw and AlphaFlow are going to be made available very soon, and these are going to be available for the public. So thank you very much for your attention, and I hope you enjoyed the talk.